Saccharides.
Saccharides, right? Monosaccharides, and we say these monosaccharides classified according to important points. Number one, the types of monosaccharides. We have different three types of, or two types even, according to the number of carbon atom, number of carbons, and position of what?
Position of the carbon.
Sorry?
Position of atoms.
Position of?
Atoms.
Which atoms you mean, atoms?
The carbon, what's it called?
Yeah. What's called? The position of?
The carbon atoms.
The carbon atom? No.
No, wait.
Which group? There's a group in the structure.
Carbonyl.
Carbonyl, excellent. The carbonyl group.
Oh, yes.
Carbonyl group, yeah. And we said, based on number of carbons, we have 3, 5, and 6 carbons. What we call the 3? Remember? Yalla. Yeah. Who said it? Yes, we just said it. Triose. Okay. Yeah.
Triose, pentose, sextose.
Pentose and sextose, yeah.
And pentose, right?
Sextose.
And hexoses. Yeah, that's correct. Pentoses and hexoses. Okay. What I want to say, yeah. Here I think is E, not I. Done. Now, and according to the position of carbonyl group, we have two types. What are them? Aldoses and what? Aldoses and ketoses, right? We say the aldoses, the position of carbonyl group, where? In the middle or in the
right.
Where is the position of? Is it in the middle, aldoses, or on the top?
Middle, I think.
No, in the top. Ketosis in the middle. Ketosis. You can memorize it. Me, as when I was a student, I memorized it by what? Ketosis, cut, so it's in the middle. You understand?
Okay.
Or between the carbons. So you can cut ketosis. Okay. So it's between the carbons, not on the residue, on the first one. Not on the first carbon. It's between the carbons. Got it?
Yeah.
Okay. And we took different examples. Then we said these monosaccharides, when they bind together, it gives disaccharides, right? And we say the disaccharides, such as glucose, when linked to another glucose, right? It gives you what? Maltose. And glucose with galactose, that gives what? Do you know how to memorize it, guys? It's easy. We have 3 main examples. You have to know it. All of them, they have glucose. MashaAllah, this information from grade 9. Okay. How to memorize it? Look. All of them start by glucose. Glucose. The 3 examples which mentioned in your book. Okay? The disaccharide. The first one, glucose with glucose. Same. The second one, glucose with galactose.
Is it lactose?
I will come to the result. Wait. And the last one, glucose with fructose. Okay? Is it clear till now? So all of them, glucose with another monomer. Okay? The glucose with the glucose, keep it to the last. I will come to the second one. Glucose with galactose. How to memorize it? Just remove gal. So lactose. Okay? It's lactose. Easy? Okay? Yeah. So glucose with galactose, just remove ga, so it becomes lactose. This is lactose. The last one, glucose with fructose. This is the fruit sugar. We called it the common name. Okay, if you want to buy it, you say, give me the fruit sugar. Okay, fruit, fruit sugar, sucrose. Okay, so, sucrose in Arabic, sukkar. So you can memorize it. Sucrose. Okay.
Yeah.
Come to the first one. Glucose with same glucose give you maltose. Remember Maltesers? You know Maltesers, this thing?
Yeah,
Yes,
yeah.
Yeah, but don't write Malteser, write maltose, okay? Yeah, so it's maltose, okay? So glucose with the glucose, maltose. Glucose with lactose, so it's a lactose. Glucose with fructose, which found in the fruit sugar, saccharose, okay? Easy to memorize it. This is the disaccharides examples. It's a two monomers linked together by what we call the link. What we call the link between them.
Nitrogen diffusion.
Those.
Glycosidic, right?
Bond.
Yeah, glycosidic bond. This is the name of bond between two monomers, two monosaccharides to form disaccharides. And we talk two types of reactions, the type of reaction used to link two monomers together. What we called it? The reaction, which we remove water or add water to link them together.
Dehydration.
Yeah,
Dehydration.
the dehydration
Two
part.
monosaccharides link them together by removal of H2O, and this is called dehydration. Yes, dehydration. Synthesis, because synthesis means building monomer, disaccharide, right?
Yeah.
That's perfect. Okay, guys. Then we move to the polysaccharides, and we said we have two important examples such as starch and glycogen. Today we want to describe the disaccharides in more details, actually, because it's important for us to understand the structure. Okay, now in before, when you were in grade 9, you talk starch and glycogen. They are used to store the glucoses or the energy in the animals and plants. Starch in plants, in animals, glycogen, right? And in grade 12, you knew that in starch we have two types, amylose and amylopectin. Due to what? That the amylose is a series of glucoses, glucose with a glucose with a glucose, series of glucoses in amylose without branches, but it's in a helical shape. Don't forget this, helical shape. While in amylopectin structure, it's branched. It has branches. Okay. Glycogen is a series of glucoses, but more and more branched. Okay, highly branched. Actually, I want to focus on this is the starch and the glycogen. Finished. Okay. These are used to store the energy. We have another examples rather than starch and glycogen. It's the cellulose, right? Cellulose is another example of polysaccharide. Okay, polysaccharide used in plants to make the cell wall. And we said last time it's a tough structure and it's undigestible in humans' gut. Okay, but in some animals' gut, it's digestible. Okay, the earthworm, for example, can digest it, can digest the cellulose, the cell wall in plant cells. But us, our guts, we cannot digest it. Today we will know why. Okay, let's see. Actually, the ability to digest cellulose or not, due to the structure of cellulose. Let's understand what is the structure or how the starch structure built up and how the cellulose structure built up. And we compare between them. You understand? The first point, what you have to know, that both of them are polymers of glucose. Means that it's a repeated glucoses linked together. But how they are different? Actually, the first point which you have to know that the glucose has two forms. The glucose has two forms. I'll go there. You have to know that the glucose has two forms, means two structures or isomers, we can say, okay? The first one, the regular one, which you know it, looks like that. Oh, where is it? What happened? Sorry. You see what I drew on the board?
Yes.
Yes, sir.
Yeah, good.
Yeah.
You know that these are carbons. Here, oxygen. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4. I don't know if it's clear for you. I'll zoom in. Carbon number 5, and you know this is glucose, C6H12O6. And here we have carbon number 6, and this carbon attached with 2 hydrogens and oxygen. Okay. Also, this carbon 1, 2, 3, 4, 1, 2, 3, 4, 4, it has hydrogens and oxygen. Not oxygen. Help me. What, I forgot the name. What the name of this? And here, OH, H, here H, OH, and there OH, H. Okay. Now, this is the structure of glucose. The first form. The second form is same. Look, no difference. Only one difference. But this difference make a change in its chemical property. Okay. Same. Oxygen here, carbon number 1, number 2, number 3, number 4, and here number 6, CH2O. Here, same, OH, look, same, no difference till now, okay? OH, H, OH, H, same. The difference actually here in the carbon number 1, what is the carbon number 1? Exactly the carbon which after oxygen, clockwise, okay? The first carbon, this carbon number 1. It's the position of OH and H. The OH up, the hydrogen downward. Okay? So if the OH in the up side or the upper side or toward the up, this is called beta glucose.
What's the other
While
one called?
alpha glucose, if the OH downward. So here, the two forms of glucose are same in everything except the position of, or the direction actually, of OH, upward or downward. If it's upward, so it's beta, up, B letter, okay? Upward, beta glucose. If the OH downward, this is alpha glucose. Got it till now? Is it clear?
Yeah.
The others, easy to memorize it, guys. Look, start alpha glucose. Just memorize alpha glucose. OH downward. The next one downward. Upward, downward. Means that all of them downward except one upward. Okay? The other one, these three are same. Downward, upward, downward. The difference only the first one, as we said. If it's upward, so beta glucose. If it's downward, alpha glucose. You have to memorize the structure of glucose. Okay, what is the importance of this? Is that effect on the how the glucoses link together, if it's alpha or beta? Yes. Let's see how two alpha glucoses link together. If you want to link two alpha glucoses together, how it links together? Wait, let me see if it's in the PowerPoint. Yeah, here. Or I will draw it, then I will back to the, where's the, do you see the board? Guys,
Yeah, we see the board.
you see the board,
Yeah, the
right?
board.
Yeah, because I see the PowerPoint. Not the... Yeah. Okay, now you see the board.
Yeah. Yeah.
Okay, let's take one alpha glucose and link it to another alpha glucose. But I want to fix something here to be easy for you. This one. I want to draw it again. I didn't change anything. I just draw it with. Okay. I'll tell you that I will take two alpha glucoses and link them together. You agree with me? Huh? I'll take this. Okay. Sorry. Wait. Come on. Okay, again. I take this one. Okay. Copy. Then come here. Paste. And I need another one. Okay. Paste. Okay, I have now two alpha glucoses, right? As you see. I'll bring them close to each other so I can link them together like this. Okay? How two glucoses link together? Actually, the bonding between two glucoses occur between carbon number 1 and carbon number 4. This is carbon number 1 and carbon number 4 on the other one. Okay, guys? How the glycosidic bond formed, as we know, by dehydration synthesis, right? And how dehydration synthesis occur? We said last time it's between the OH, right? This OH and this OH, they do reaction together. How? By removal of OH from here out and removal hydrogen from this OH. You got the point? I will repeat again. I have here OH, OH. How glycosidic or how dehydration synthesis occur, how dehydration reaction occur. The OH here out and the hydrogen here out. So what left? Link.
Oxygen.
Oxygen, link.
And carbon.
You got the point? And carbon. So carbon, oxygen, carbon. So the bond will be like this. Okay? Carbon, oxygen, carbon. You got the point? Is it easy? So it's like this. Okay? Because the OH cancelled and the hydrogen from here cancelled. This is the glycosidic bond. Glycosidic bond. You have to notice important point that the OH and the OH in the two glucoses are in the same, they are matching together. That's why they link together on this way. They react together. Okay? And this is happened by using enzyme, of course. Okay? Enzyme to do this reaction. Okay. What we call this bond? We called it alpha because used alpha glucose with alpha glucose. 1 means carbon number 1 to 4 means carbon number 4 glycosidic bond. You got the point, guys? So
Wait,
alpha,
sir, can you repeat that?
again, the name alpha because we use 2 glucoses alpha. The 2 glucoses alpha. 1 to 4 means the bond between carbon number 1 and carbon number 4. So alpha 1 to 4.
Okay.
Okay? Okay. Got the point?
Yeah. Yes, sir.
Very good. Right. Let's see what happens if we want to bond 2 beta glucoses together. We know that beta glucose is same except the direction of OH in number 1 only. Okay? It's up, beta. So OH up. So easy to draw it. Do you see? This is the oxygen. Here, hydrogen. Here, we said downward, OH. H. Here, OH. Sorry. H. Okay. And the last one, it's no change. Downward. Same. Okay. This is the CH2O. Okay. Remember, carbon number 1, only the different one. It's upward. This is what beta glucose. Carbon number 2, no change. Number 3, no change. Number 4, no change. Type. If we want to link 2 beta glucoses together, let's take a copy from here. Copy, paste. Okay. Now I have here another one, copy of beta glucose. Okay, guys? Bring it close to the first one. Oh, we have a problem here. Who knows what is the problem now?
It's not inverted.
Yes.
Yeah.
Yes, huh? The OH here and the
It's on the
OH
other
here. Yeah, so we have problem. They cannot now link together due to what? The position. Time. Who can suggest a solution for that?
Just replace it.
Replace it by what? Replace the OH and the H?
No, sir. Cancel the oxygen with each other.
Sorry?
Yeah.
Sorry?
Sir, do you mean like out of the question or?
No, I say suggest how to solve this problem. Now the problem, the OH in the second glucose is not matching with the OH in the first glucose. We said,
Yeah, sir, just
guys,
replace it.
we said the glycosidic bond, it's between carbon number 1, carbon number 4, okay? Because they are beside each other. And the OH linked
We
to
can
OH
bond carbon
with,
we can link. No, wait, no.
maybe some of you say, why not this and not this linked together? No, can't.
Yeah, no.
Okay? Can't. So we need to find a way to make the OH of the first glucose link with the OH of the second one. Easy, ya shabab.
Delete or what?
Do we just move it up?
Sorry?
Do we just move the other glucose molecule so they're on the same level?
No. What do you mean by that? Type. What if? What
Can't
if?
you just cancel them both out?
Sorry. What you said, Majid?
No, I said, can't you just cancel them both out?
Cancel what? No.
Never
you can't.
mind. No,
No. You can't. You can't. They have to be on the
Okay.
same direction. Actually, here, what if we flip the second one upside down?
Sir, Wallah, I was about to say that.
Yeah, why didn't say?
Mr. Me
Right.
Too, I was about to say that.
Let's try. I will draw. I will draw it upside down to see how it seems. Wait.
Well, didn't I say in the first place it's inverted?
Inverted, but you didn't mention exactly. I thought you mean inverted H and OH on carbon number 4. That's what you mean, by the way. Right?
Oh, okay.
Yeah. But Here I'm saying invert all the molecule, flip it upside down. Means that, do you see this CH2OH? Now it's
It will
downward.
become down.
Yeah, downward. Okay, and this is the oxygen. Okay, and here now the H up, the OH down. Oh, and here the OH up and the H down, and here H, OH, OH, H. Guys, there's important point here. Sometimes in the question, if I give you this structure, the second one, and ask you this is beta or alpha, and some of you, what will do? They will look at this is carbon number 1. They think that this is carbon number 1, and they say, okay, it's downward, so it's alpha. This is wrong. It's still beta, because I flipped only the molecule. I didn't change the position of OH and H. Okay? The OH is still upward. Look, it's toward the oxygen. Okay, toward the oxygen. It just flipped upside down. Okay?
So I have a question.
Yes.
How would you number the carbons when it's upside down?
In chemistry, we know they said you have to count clockwise after the different atom. Here we have only the oxygen different atom. Okay? And the others carbons, right? So how to know what is the number of this carbon? Move clockwise after oxygen in the right side. Okay? So this is oxygen number 1. You got the point? So here, this is the oxygen. Now it's flipped upside down. So move to the right of oxygen. This is the move like this. Okay? Sorry, like this, because it's upside down. So it's not clockwise now. It's opposite. Okay? So this is number 1, number 2, number 3, number 4.
Okay.
Okay? You can imagine it. Just flip it. Okay?
Just anti-clockwise.
Yeah, exactly. Now, so is it so what the benefit of this? Let's see. If we cut that one, and take it away, bring the other one, which is flipped upside down as we said, and fix it on its place. Oh, what now? What do you see?
We can we can bond them by
You can merge
dehydration.
it.
Yeah, now we
Bond it.
can, yeah, wait, I want to take this away. This is the normal one, not the flipped, right? Okay, I'll take it here. Done. Now, this OH and this OH are matching in front of each other. So linking is, or bonding occur. The OH cancel, the H cancel. So the bond will be O, but upward. Look, yani it's, sorry.
Yeah.
It's toward up like this. Like this, like this. Okay, and here oxygen. You got the point?
Yeah.
Type. What if we bring another one beside it? Let's see, I will give you 2 examples of chain of alpha glucoses and chain of beta glucoses. What is it?
So what's the bond called between this?
Glycosidic bond. Oh, yeah.
No.
Beta 1 to 4 glycosidic bond.
Okay.
Okay, because it's same. I just flip it upside down. I'll back to it here. What is it? Yeah, that one, right? That one. It's 1 to 4, but beta, we say. It's a beta.
Okay.
So this is the 2 ways how glucose is linked together based on its type, alpha or beta. We can see these chains. Now tell me, this is a starch chain. Is it alpha or beta chain?
Alpha.
Alpha.
Oxygen is on top.
Alpha. Oxygen is up, up, up. Excellent. Okay?
Yeah.
So this is alpha 1 to 4 glycosidic bond. Okay. What about the next one? The next one.
I can't see it. Okay, there you go.
Now here.
It's alpha.
No,
It's
it's beta. It's beta.
alpha or beta?
Beta, because it's alternating. It's up and then
Yeah.
down, up and then down.
Look at the second
Beta.
molecule. The second molecule.
Yeah, it's not straight.
Yeah, the second molecule flipped upside down. Now, if you want to
Yeah.
link the third one, you put it regular beta, not flip. Okay? And the next one, flip.
Okay, flip, flip, flip.
This one, regular, flip, regular, and so on.
Yeah.
Okay? So this is what? Beta or alpha glycosidic bond?
Beta.
Beta.
Beta
Beta.
glycosidic bond. Also, it's 1 to 4. I forgot to write 1 to 4. Because between carbon number 1 and carbon number 4. Is it clear?
Yes.
Okay, guys, time is going to finish, so we need to open again another class. Sorry, because the account is not upgraded yet. Yeah,
Okay.
I'm waiting to rejoin, okay?
Okay.
Yeah.